Up until this point, up until recently, I think the most major need was an analysis of formalin-fixed samples. Whole genome sequencing was put forward as an answer to it, but to be honest, the trials of whole genome sequencing and formalin-fixed have been disappointing up to the point. There are still technical difficulties with the library prepreparation, and it's a bit hit and miss. Only about 1 in 3 to half of the samples put forward for whole genome sequencing from FFP work properly, whereas the development of exome sequencing methodologies meant that we can access most of the clinical trial samples, and our success rate is over 95%, and this is reproduced by others. So this is definitely an advance. Other things we need to go forward into the future, single cell sequencing is a big popular topic at the moment, and understanding how we can use single cell sequencing in clinical oncology projects is going to be particularly important. At the moment we can do it in fresh tumours, but they need to be live, i.e. just come out of the patients. So we need to move towards methodologies where we can do single cell sequencing on either tumours that have been frozen down and then cells extracted, or even better on FFPE samples where we can take a couple of sections, dissociate into a single cell suspension and sequence it. And the reason it's important is we'll be able to then understand things like tumour heterogeneity, different subtypes within a tumour, and molecular response to agents, which might explain some of the differential response you see, especially in metastatic cancer.